as a member on the Arthritis Foundation OA Clinical Trial Network Steering Committee, I would like to use this as an opportunity to introduce a network to the community very briefly. So as Dr. Steve Taylor mentioned at the very beginning of the workshop, that the Arthritis Foundation OA Clinical Trial Network was established in 2017 with a goal to establish an infrastructure and basically a network that can help to accelerate future OA drug trials with sites that are ready to recruit patients and collect data. And currently, this network is spearheaded by Dr. Jason Kim from Arthritis Foundation and leaders in the academic and the clinical fields, including Dr. Martin Lowe's and David Felsen. And we have members of leading clinicians and researchers in the field. So the network expanded from 4 sites at the beginning to 12 sites now, with more than 70 members today. And the network has been focused on the groundwork, including the multi-vendor, multi-site cross-validation of cardiac T1 T2 imaging, as Shamil mentioned very briefly, and the biomarker measures to establish sites, again, which are ready for patient recruitment, as well as to collect a high-quality data for both biospecimen and quantitative imaging. So the network currently has sponsored several cohort studies and trials, including PTOA for the patient who had the PTOA and would like to start performing clinical trials on OA novel drugs, hopefully very soon. So with that, if I may, I would also like to make a few quick comments as an imaging researcher focusing on quantitative imaging development for OA. I would like to echo what Shamila mentioned very briefly in her talk, that the standardization of imaging measures for both acquisition and analysis of post-processing and to evaluate the inter-vendor inter-site variation are critical. And along this line, both KIBA MSK committee, co-chaired by Dr. Thomas Link from USSF and myself, and the AF-OA Clinical Trial Network have made efforts towards this direction. So with a sponsor from Arthritis Foundation and NIH, we now have the cross-validated T1 raw and T2 imaging on all major MR vendor systems. And the KIBA MSK committee has also worked on the profile with recommendations of T1 raw T2 imaging acquisition and processing method. And in addition, that with AI technique, we have seen that we don't need people to do the tedious manual segmentation and processing, and the automatic method is more consistent and reproducible. So I really think we are probably at a prime time that we can potentially apply this quantitative imaging method in large clinical trials. But I think there are still some roadblocks that I would like to mention. One is, for example, we would need help from our industrial colleagues and partners to have these advanced techniques, for example, T1 imaging as a product sequence, so they can be used in large clinical trials instead of just within a few academic centers. And or we would like to know what information or what we can do to help our industry partner would need from us in order for them to move forward. And secondly is regarding back to this as along the line to have quantitative imaging as a potential surrogate marker, we talk about the data reproducibility, discrimination ability, as summarized by very 2 recent nice systematic review and meta-analysis by McKay and Arkinson, respectively, but they are limited to cross-sectional evaluation. So I feel as a field, probably it would be helpful if we at least begin to work together, maybe have another systematic review on so far the longitudinal study for T1 and T2, and to see what we have done regarding their prediction ability to cartilage loss or pain or other clinical endpoints along the line, again, to potentially have this imaging measures as a potential surrogate biomarker, and then we'll see what we have done and what we need to be done, so we can move forward as a field. So those are 2 quick comments I would like to make. Thank you.